In section 3 of our chapter on carbohydrates and nucleic acids, we're going to discuss how Fischer determined the structure of the basic sugars up to 6 carbons in length. Fischer discovered these structures by synthesizing, or essentially building, using chemical reactions, the sugar structures. This synthesis is called the Keeling-Fischer synthesis of sugars. Fischer won the Nobel Prize in the early 1900s for this work. He studied the sugars by making them using this reaction sequence. The sequence can be summarized as follows. It starts with an aldehyde sugar, an aldose sugar. It makes the sugar 1 carbon longer by adding a cyanide to the aldehyde in a reaction known as the cyanohydrin reaction. We discussed the cyanohydrin reaction in our chapter on aldehydes and ketones. We will later see that it is possible to convert the cyano group, which is often also called a nitrile group, into an aldehyde. When the cyanohydrin adds to the aldehyde, it creates a new chirality center on the carbon that was the aldehyde. Because the aldehyde is planar, a 50-50 mixture of both configurations is made in the product. This is easiest explained using a diagram. We're going to start with D-glyceraldehyde, just as Fischer did. And we're going to imagine adding a cyanide to that carbon. Now, a couple things about this. First of all, when the cyanide adds to the aldehyde carbon, to make room for that new bond, a pair of electrons will be pushed out onto the oxygen. In a subsequent step, the oxygen would become protonated. So the carbonyl oxygen would become a hydroxyl group. This would also create a chirality center because we would now have 4 different groups attached to what had been the aldehyde carbon, indicated here. Conceptually, there are several ways to imagine this. What I've tried to do is imagine building the cyanohydrin product in the same orientation that it would be in a Fischer projection. So in this case, I imagine what would happen if the cyanide group always added from the back to put it vertically on a vertical bond, which would then be dashed. And to create the 2 chirality centers, what I imagine is that the aldehyde is in the plane of the paper, but that it exists in one of 2 possible rotations. One where the carbonyl is on the left, or rotating 180 degrees around this bond, the carbonyl group would then be on the right. In this case, effectively then, if we always add from behind the plane in the same orientation, we would be adding from one face or the other face of the aldehyde. We can then leave the hydroxyl group in its orientation relative to the chain of the Fischer projection. Thus, in the top one, the carbonyl oxygen would become an OH on the left-hand side of the chain. And on the bottom structure, it would become an OH on the right-hand side of the chain. So we can see that we create 2 diastereomers because the lower numbered chirality center does not change configuration, but we create a 50-50 mixture of the 2 configurations of the newly created chirality center. We can then do a reaction to convert the nitrile into an aldehyde. We're not going to cover the specifics of this reaction in this chapter. I just want to point out that one way that this can be done is by adding hydrogen with a palladium catalyst and then hydrolyzing. But conceptually, we can imagine that there are ways to convert this nitrile into an aldehyde, thus creating overall a sugar molecule that is 1 carbon longer in a 50-50 mixture of diastereomers. You can then imagine taking each of these molecules, adding another cyanide group, creating 2 more diastereomers here, 2 more diastereomers there. So there should be a total of 4 5-carbon sugars. It should also be pointed out that because the chirality center, which we got from D-glyceraldehyde, is not changed in the subsequent products, every sugar that would be synthesized through this cycle would have the D configuration. We can summarize Fisher's work by looking at the chart on the next page. This shows all of the D aldoses up to 6 carbons that were synthesized and characterized by Fisher using this method. I should point out that the plus and the minus in the names indicate the direction that the sugar rotates polarized light. It's determined experimentally, and the important thing is that each enantiomer will either be a plus or a minus, but the diastereomers each can have their own unique relationship of the enantiomer to the direction that light is rotated. Let's take a look then at this chart. You can see that we start from D-glyceraldehyde because we make 1 additional chirality center in a 50-50 mixture of configurations that creates 2 diastereomers of the 4-carbon sugar. Then each of those 2 diastereomers creates 2 diastereomers of the 5-carbon sugar for a total of 4 5-carbon sugars. And then again, each of those diastereomers of the 5-carbon sugar creates 2 diastereomers of the 6-carbon sugars for a total of 8 6-carbon sugars. If you look at these, you will recognize some of the names of these sugars. So for example, I've already mentioned glyceraldehyde. Here's ribose. Here's glucose. It turns out that several others occur less importantly in biological chemistry. So for example, galactose is a component of the disaccharide sugar lactose. In theory, you could be required to learn all the common names of all the aldoses shown on the previous chart. In practice, this is really not necessary. Most of those aldoses are not chemically or biologically significant. Therefore, in this class, you will only be required to memorize the following 4 monosaccharides. These are by far the most significant monosaccharide sugars that are found in biology and especially biochemistry. The 4 that you need to memorize are these: D-glucose. D-glucose can be remembered because it has a total of 6 carbons. The top carbon and the bottom carbon are not chirality centers, so they are not on crosses in the Fisher projection. The remaining 4 chirality centers have the OHs on right, then left, then right, then right. Right, left, right, right. I actually memorized that acronym to be able to draw from memory totally correctly every time the structure, the Fisher projection of D-glucose. Related to D-glucose is D-fructose. D-fructose is a ketose. It's a ketone sugar. Conceptually, it's a sugar where the carbonyl C double bond O has been moved from carbon 1 to carbon 2. And in carbon 1, instead, we have just an OH attached. If you look then, what this does is it eliminates one of the chirality centers in D-glucose because the carbonyl is trigonal planar. It's no longer a chirality center. However, interestingly, if you look carefully, the remaining 3 chirality centers in D-fructose have the same configuration as the corresponding 3 chirality centers in D-glucose. This implies that D-fructose is made from D-glucose, and in fact, there exists a reaction which will do this, which we will discuss when we cover biochemistry later this semester. D-ribose is a 5-carbon carbohydrate. It has all of its OH groups on the same side. In the D form, that means right, right, right. D-ribose is important for a couple different reasons. The first of which is that D-ribose is part of the building blocks of nucleic acids, DNA and RNA. We will see that later this section. It is also very important in photosynthesis to create D-glucose. Finally then, we have D-glyceraldehyde, which again we saw as part of the Kiliana-Fischer synthesis. But it turns out D-glyceraldehyde in a slightly modified form with a phosphate attached is a very important component in what is known as glycolysis, which is an extremely important biochemical metabolic pathway that we will talk about in a later chapter.